I've started recording. So we've still got a bunch of people piling in, it looks like.
Well, Natalie, what do you think? I cannot see the activity of people joining, but I'd say let's go ahead and get started.
We've got 22 people in, including ourselves. So looks like we're probably good to go.
Okay, why don't you kick things off and we'll get rolling.
Okay. So what we're gonna do, the way we generally do this, is we'll have Jeff and Steve give the presentation. I am going to be muting people throughout, so just so that it's less confusing, so that everybody can hear. If you have any questions either during or after the presentation, please feel free to put them into the chat bar on the side of the GoToMeeting control bar. If it's an easy question, if it's a quick question, I can answer it, and I'll be watching it during the presentation. If it's something that I think would be useful for Steve or Jeff to elaborate on, then we'll hold it and we'll have a question and answer session at the end. So any different or any questions anybody has, please feel free to put them in the chat bar. And with that, Jeff, go ahead and get us started.
I will do so. Thanks, Natalie. Thank you, everyone, for joining us. My name is Jeff Farmer. I'm the CEO of Diagnosis. And again, thanks for joining us. Just to reiterate one thing that Natalie mentioned, questions are highly welcome. The chat bar is the easiest, and we've set up the presentation to hopefully allow plenty of time for discussion through chat, and then we'll address questions at the end. So please send them in to us. So the agenda for today, topics, I will do a short introduction to Solaris and go through some of the basics of the types of electrophysiology tests that we can do on rodents on Solaris. In addition to myself, Natalie will help us run the session, and we have many people from Diagnosis joining the call, so that will help facilitate if there's follow-up questions and discussion that you'd like to have. Our team or Dr. Nusinowitz would be happy to do that after the call as well. After my slides, I'm pleased to have Dr. Steve Nusinowitz join us for this webinar. Dr. Nusinowitz is a visual physiologist in the Department of Ophthalmology at the Stein Eye Institute, where he serves as the director of the Visual Physiology Lab for clinical studies and is director of live imaging and functional evaluation core for animal studies. So he is responsible at Stein Eye both for the clinical electrophysiology work as well as the preclinical electrophysiology work, along with other duties and responsibilities that he has. So it's a great honor and joy to have Steve join us today. So I'll try and get through my slides quickly, and then Steve has 3 very interesting cases that he'll take us through today. So just a couple of quick introduction slides for those of you that may not know too much about Diagnosis. We were founded in 1984. We have offices just outside of Boston in the United States and just outside of Cambridge in the UK. And we're privileged to have distributors around the world. We have about at least 15 distributors working with us in countries outside of North America and Europe. Historically, we've focused on providing electrophysiology systems, both human and animal, to large clinical and research institutions worldwide. Our first customer was one of those institutions here in Boston back 35 years ago, and that's been the primary focus of the company ever since. We also work a lot with pharmaceutical and biotech companies in clinical trials. Many ERG systems from diagnosis out in the field, and again, one of the joys of being part of diagnosis is working with our customers and supporting their research and helping them with that in any way we can, beyond just the equipment, any support and services, and ultimately that teamwork has led to many hundreds of peer-reviewed papers that have been published over the years using diagnosis systems. Just one slide on the full set of systems that we have for animal testing. We'll talk the rest of the presentation about Solaris, but before we do that, we have a set of equipment actually that covers anywhere from very small mammals, which is what Solaris is designed for, rodents, guinea pigs, rabbits, etc., up through larger mammals and primates at the bottom of the page. It's basically our human clinical systems. We have many customers that use those for larger mammal testing. And then our initial revision 1 mammal test platform, the ColorDome Lab Cradle, we still do sell. Pretty much Solaris has replaced that. There are a couple of niche applications like zebrafish that people still use the ColorDome for as well. So just a couple of slides then on Solaris as a warm-up for the cases we'll talk about today. Solaris was launched about a little over 4 years ago now. It has a couple of big advantages that we tried to bring into this platform compared to our legacy platform, the Lab Cradle ColorDome. One is its compact size. It's 41 by 18 by 13 centimeters. The picture shows the entire system along with a computer. The systems can ship customer choice either with a touchscreen laptop or a touchscreen desktop. Easy to train. So one thing that we had intended, and it seems to be true, is new users or users new to electrophysiology are able to pick up using the system quite quickly. For those of you that do electrophysiology, know that it's a subtle process and technique to do it well. And Solaris is intended to help serve you in that process. And about 50% of our sales in the US so far have been to customers who had not previously done ERG testing. So outside of ophthalmology, many neurologists and other medical groups have been able to pick up Solaris pretty quickly. Rapid setup of animals. So depending on your protocol, the whole intent of Solaris is to enable you to set up your animal very quickly, and we'll talk a little bit more about that. But it offers the possibility of processing many mice, 10 or more per hour, again, depending on your protocol, but at the same time has very high performance. And both of those attributes come from the design of the electrodes and the stimulators, which we'll go through. Broad set of tests. So nearly all electrophysiology tests that you would want to do on a small mammal, a rodent, can be done on Solaris. So we'll go through those. One of the key attributes of the design is the fact that we integrated the electrodes into the stimulators. So on the right-hand side of the chart, these are the full field stimulator and electrodes. You can see the person's hand in the top picture just for scale. That person is holding on to the stimulator itself. The LEDs are embedded in that stimulator. There's a light pipe then that guides the light from the LEDs, which are at the back of that device, down the light pipe and out a cup-shaped optic at the end of the stimulator. And so the light goes directly into the pupil of the animal. And then, very hard to see in these pictures, but the red arrows point to the silver chloride electrodes. Again, those are hardened electrodes that are manufactured directly onto the stimulator. So the whole device is designed to last for many, many years, and the electrodes are automatically placed as you place the stimulator. The electrodes, similar to processes you would use on other systems, do require an ophthalmic gel then to provide electrical contact between the cornea and the electrode itself. But the idea of the electrodes is to make them more durable than other electrodes have been in the past, faster to place, And then the circular architecture makes them a little bit more tolerant to placement on the eye, which helps the speed of testing and repeatability of the testing. So we have both full field, and then over on the left-hand side, a major innovation of Solaris is integrating a pattern stimulator into this same architecture. So that's literally a little, you can think of it as TV monitor on the left-hand side. It's an OLED monitor sitting behind the optic and the electrode. System features. So one of probably the major benefit of Solaris is what we call touch-touch protocols. So most of the protocols on Solaris we're able to do using just the electrodes that are in the light guide electrode devices and avoid using needle electrodes. Needle electrodes are possible, and we have more than enough ports in the amplifier for Solaris to accept needle electrodes. But if you can avoid using needle electrodes, it speeds up the setup of the animal, and we found actually increases the performance, because in that case, you're stimulating one eye, say the right eye, you're using the left eye as a reference, and the fellow eye in both animals and humans has been found to be a very good reference location. So our touch-touch protocols I'll show in a second are a major thing that you can do when you bring the light stimulus right up to the eye. Our software has the ability to customize protocols very quickly, and then there's multiple different ways to export the data. You can print, you can transfer data, you can export raw data, and you can mass export data as well. The other thing I would point out, this is important as well, is the heating element to keep the rodents warm during test at about 37, 38 degrees Celsius is integrated right into the console of Solaris, along with the amplifier and the control electronics. So all of that's designed right into the console, enables the system to be very low noise and convenient. It takes a couple of minutes once you turn on the system for it to warm up to temperature. Then you've got an automatic heating plate to keep the animals at the proper temperature during test. This is a video then, I'll just start it and hopefully this comes through for everyone. This is a touch-touch flicker protocol. So again, no reference electrodes, no needle electrodes. In this case, hopefully you can see the right eye is being stimulated and now the left eye is being stimulated. So at this time, the electrode on the left eye is being used as the active reference, active electrode, and the right eye is the reference electrode. And our software can automatically flip back and forth between the two. So quick setup, fast test. Again, you can do a more traditional setup with a needle electrode as a reference and stimulating both eyes. Any of that is possible, but the touch-touch protocol is something that's a little unique to Solaris itself. So I'll go through just a little bit of warm-up data here before I turn things over to Dr. Nusinowitz. So this test on the left-hand side shows some baseline tests that we've done looking at stimulator and electrode position. So basically the system is set up to enable you by hand to bring the electrodes and the stimulators up to the eye itself. And we've looked at the tolerance of test results in slightly mispositioning the electrodes. And you can see on the left-hand side that as long as the electrode is in good contact with the cornea, you should expect to get good repeatability from the system. And on the right-hand side, that shows it as well for just some basic ERG tests. A couple of charts then to go through a full set of tests that the system can do. And these are not quite even all of them, but just to give you a flavor for the tests that customers have done on Solaris to date. So starting in the upper left-hand corner, these are traditional dark adapted ERG tests that enables you to mark and measure the function of rods, bipolar cells, and then the oscillatory potentials give you a measure of function for the amacrine cells. Working my way over to the right, that's a C wave test. So the A and the B wave are just over on the left-hand side, that's the y-axis. The C wave is used primarily to measure the function of the retinal pigment epithelium. Over on the far right-hand side, light-adapted ERG and flicker tests. And then going along the bottom, with our dim stimulators on Solaris, you can do a scotopic threshold response. So that typically would be a whole series of flashes. I've just pulled out one where the B wave is just starting to show up. But again, typically the protocol is set up in about 10 or so steps to measure the scotopic threshold and then look at the B wave and the A wave at higher flashes. The system also does a flash VEP, so that's typically used to measure the function of the full visual pathway to the cortex. And then a less used protocol, but one that runs just fine on Solaris, is called the PHNR, the photopic negative response. And for users who are doing PHNR tests, they're typically looking at the function of retinal ganglion cells. Those are all tests that can be done on the full field flash stimulators. On the next page, then, these are some examples of tests that can be done on the pattern stimulators. The two most commonly, and probably first the most commonly used with our pattern stimulators, is what's called a pattern ERG, or a PERG. Through decades of research, that has been shown to be a good test to measure the function of the retinal ganglion cells. So you can see PERG examples here from 3 different animals: a mouse, a rat, and then a guinea pig. On the bottom, pattern VEP. So basically the same pattern stimulus can be run on the monitor, and then electrodes are placed near the visual cortex and at the front of the animal as a reference, and pattern VEPs can be measured on Solaris as well using the pattern stimulator. Those are both protocols that are validated and released and come with the Solaris if you buy a pattern stimulator with your system. We're always doing R&D and working with customers such as Dr. Nusinowitz on new techniques. One of those is multifocal ERG. So Steve had published a paper 15, it might be 20 years ago now, on the multifocal technique in rodents. So we've been working with Dr. Nusinowitz to implement that on Solaris as well. The COVID pandemic has put that a little bit on hold for a few months, but I think hopefully later this year we should be able to release that protocol once we fully develop the process for it. And so with that, I will turn things over to Dr. Nusinowitz. Steve, it's all yours.
Thank you, Jeff, and thanks for inviting me to this webinar. And Jeff asked if I could present some cases where we're using Solaris. We got the Solaris instrument about a year ago, and we're diligently trying to integrate until COVID hit. So we've been shut down for a long time. We just sort of came back a little bit, but the news I'm hearing today is we're headed for another shutdown. So it's not good. But anyway, I will give you some idea of what we're doing. So I run the live imaging and functional evaluation core at the Stein Institute. It's a pretty diverse group of scientists, all of whom have a long-standing tradition of evaluating mouse models using electrophysiology, and that's done in the core. So given the nature of our basic science division, much of our work is focused on the outer retina, and so the flash ERG has been a major workhorse for us, and we've looked at over the last 20, 25 years at almost every animal model that you are familiar with. And I just listed just a few of them that came to my head. RDS is retinal degeneration slow. STRA6 is a sort of a tap for vitamin A. ABCA4, you know, is a model for Stargardt's disease, for those of you who might be interested in Stargardt's. ELOV4 is the dominant form of Stargardt's, and so on. GNAT1 and GNAT2, we're working on a lot with these to test some of our protocols and to evaluate some of the contributions from rods and cones. GNAT2 is an animal that doesn't have, has a defect in cone transducence, so it doesn't have a cone signal. And GNAT1 is the opposite. So this one is a model of achromatopsia. The GNAT1 is an RP model. So much of our work is in the outer retina. Much of my work is out in the outer retina. And I'll show you some of that. But we also have a pretty significant division, a glaucoma division, which has research interests, as well as other individuals who are interested in demyelinating diseases like optic neuritis and things of that sort. But some of the things that we're working on now, and I'll show you a little data on the RBFOX2 in the next couple of slides. In addition, we've been working on visual pathway disorders like ocular albinism, the OA1 gene that is responsible for, in part, for ocular albinism. And for these studies, we've been using heavily the VEP and pattern ERGs, and they have been invaluable. We also, for those who are doing therapeutic interventions, the Life Core has been a first station to evaluate the effects of any therapeutic intervention that comes down the pike, including small molecules that are delivered to the retina, any kind of viral vectors, stem cell derived cells like RPE cells, in addition to pharmacological interventions that are delivered either intravitreally or in the subretinal space. So let me go to the next slide. Okay, just for those who are new at this, and I don't know what your level of sophistication is, but here's basically an ERG response, and Jeff had showed you this earlier. We can dissect out the components of the flash ERG to reveal function of the photoreceptor, which is defined by the A wave. And basically the A wave, there's a phototransduction cascade that is initiated when a photon of light is absorbed by a rhodopsin molecule or any pigment molecule, and it goes through a series of steps, which ultimately closes the outer segment channels in the outer segment, and this hyperpolarizes cell, which modulates then signal to downstream cells mediated by on bipolar cells mostly, and possibly also Mueller cells. You can see here on the leading edge of the B wave several high frequency oscillatory peaks, which are called oscillatory potentials. And as Jeff indicated, these are generated mostly by endocrines, at least that's the thinking, plus possibly also interplexiform layer cells. So let me try to go to the next one. Here is an example of what would be a standard protocol for us to evaluate retinal function in basically any animal model that comes down. This is our screening protocol. We run an intensity series from very low levels of light to very high levels of light, using a short wavelength stimulus that's designed to stimulate maximally the rods or the rod system. And you can see that as you increase the intensity, not only does the signal get bigger, but it also gets faster. And one of the things that we do very quickly, it's all sort of automated, and we're integrating this whole protocol into the Solaris system, is plot an intensity response function for the B wave. We can do also the same thing for the A wave, but in this instance, I'm showing you the B wave. And as you can see, it's the responses increase and then saturate and stabilize. You can fit the response functions with Naka-Rushton function, which is equivalent to Michaelis-Menten, to get parameters such as Vmax, which indicates the maximum responsivity of that retina, and K, which is the semi-saturation intensity, gives you a measure of sensitivity. Other measures include the threshold intensity. On the right-hand side is a series in which we look at cone function. After we dark adapt animals and do this testing protocol, we light adapt them for 10 minutes. This is standard, everybody's doing this. And then we measure the response to increasing intensities using a white flash. And again, here you can see that the B wave gets bigger, but unlike the rod response, it doesn't get faster. And there are various ways of summarizing the response functions. Instead of using a single stimulus to describe function, you can summarize the entire series using, for example, the Michaelis-Menten or the Nakarashtian function. So what I'd like to do is just show you three examples of where we started using the Solaris, but were interrupted by this lovely COVID thing that we had going on. And so in this first project, is on retinopathy of prematurity. Some of you know that retinopathy of prematurity, or ROP, is the leading cause of blindness in young children. And basically, these kids who are these neonates require high oxygen exposures to manage their lung immaturity, and this hyperoxia leads to delayed physiological retinal vascular development, which results in subsequent local hypoxia. And once you have this hypoxic situation, you get a vasoproliferation in these animals, and that's what ROP is all about. So we have a model, and this is all done by Alison Chu, MD, in our pediatric division. She is looking at an upstream regulator of VEGF, vascular endothelial growth factor, and I'm going to call it protein X because it is not published yet, and it will be soon. We're looking at its neuro contribution to vascular signaling in a murine and a mouse model of oxygen-induced retinopathy, which is a model of retinopathy of prematurity. The novel observation that she has made is that this protein expression is induced in the neural retinal layers under pathological conditions of oxygen-induced retinopathy, and it's hypothesized to be primarily driven by rod photoreceptors, but we know very little about its role in the development of the retina. So again, one of the first things that we want to do is look at these animals who are knockouts of this protein and see how they compare to wild type animals. So this is a project that we did with the Solaris, or we're doing with the Solaris. It's very early in its stages, on hold now. So I showed you our standard protocol intensity series that we had run on our other equipment. This is what you get with the Solaris. And what I'm plotting here are the intensity response function for a wild type animal, for the right eye, and for the left eye. And I want to point out to you how symmetrical these responses are. This is really important. If you do these studies carefully, you'll get this kind of symmetry. And for those of you who are doing experiments in which one eye is a control eye and the other eye is a treated eye, then this symmetry is, you have to have this kind of symmetry, and it gives you confidence in the outcome. Down below are examples of the animals who are the knockouts, again, right eye and left eye. And, you know, just on first blush, you can look at it and you can see that these responses are somewhat lower than in the wild type. On the right-hand side, I'm plotting the intensity response functions. I have not yet fit the Michaelis-Menten or the Nakarashtian to these to get the Vmax or the sensitivity values, but you can clearly see that the knockout animal has a lower response amplitude than does the wild type. So we can, what we also intend to do, and this follows some work that we did a long time ago, in which we're looking at the phototransduction cascade in these animals to see whether or not there's something going on at the different steps in the phototransduction cascade. And what we're showing here, these are the A waves to very high intensities, much higher than what I showed you for the testing protocol, our standard testing protocol. We need higher intensities in order to saturate the A wave. And then we can fit a mathematical model. I'm showing it up here. Don't worry about the details of this, but basically if you fit the model to the leading edge, you can get a couple of parameters. RMP3 is related to the is the maximal response that you get from the photoreceptors, and it's related to the number of, basically the number of gated channels in the outer segments that remain. So if you ran a lawnmower across the retina and cut it in half, RMP3 would be reduced because there are fewer channels. S is a sensitivity parameter that's related to amplification kinetics. So we made a first effort at this, and here are the A waves using the Solaris. They're good quality, and we intend to do this kind of a fit to see if there are any abnormalities in the phototransduction cascade in these animals. I want you to note that this is a really neat experiment, because at these high intensities, there's virtually no cone response. So these are isolated rod responses in the mouse. Okay, these are examples of the light adapted cone ERGs from our standard series. Again, they're very symmetrical between eyes, and these are somewhat lower. This is a first pass at some of the results. I remind you again of what the waveform looks like. Here we're looking at sort of upside down the maximal A wave response. This is the photo response. In these animals, you can see that they're a little bit lower, not much, but a little bit lower. There's something going on. We need to see whether this gets enhanced or washed out as we add more of these knockout animals. Correspondingly, whatever happens in the photoreceptor is carried downstream, and you can see that there's a little bit a lower amplitude on the Vmax in these animals. Okay, so what are some of the questions we're going to ask? And we're going to use the ERG as one of the assays to evaluate this. Are there developmental changes in these animals that have this knockout? Are there retinal effects associated with protein X expression levels? And we can modulate the expression levels in these animals and see what the consequence is on the physiology. There's also a therapeutic intervention in the works, and that's why I can't tell you what this is. Sorry. And we're going to see whether or not this intervention mitigates not only the vascular problems, but also any retinal issues that we might find. And also, if you vary the concentration levels, how does that reflect in the ERG and the vascular issues? So that's one project. Can I go on to the next one?
Yes.
Okay, in the second one, we're moving a little bit away from the flash ERG. This is a project that was brought to us from our glaucoma division, Natik Piri, who is one of the basic scientists in the glaucoma division. And he is working on this gene called RBFOX2, which is expressed in the retina, mostly restricted to retinal ganglion cells, amacrine cells, and horizontal cells. And just to give you a little background, the RBFOX2 is a master regulator of RNA splicing and is known to be involved in synaptic transmission. And what we're trying to do in this project for them is to determine by ERG and pattern ERG whether or not the RBFOX2 up or down regulation, and we can do that with, he can do this with some of the vectors he has, affects retinal ganglion cell function and possibly also amacrine cell function. So the basic protocol for this study, the focus is on the modulation of retinal ganglion cell function. So PERG is the most informative. However, we also require standard flash ERGs because we want to assess what this mild labeling in the outer retina is having. RBFOX labels in the outer retina a little bit. We also need to get at the flash OPs, and that's why we have to add a flash ERG. And more importantly, we need to sort of see how signals are being transmitted from the outer retina through the system. So we need to know what's going on in the outer retina before we look at the ganglion cell. So here is an example of a readout for these animals. Again, there's our standard protocol, dark adapted ERGs. Again, look how beautifully symmetrical they are with these with this particular animal. And I kind of attribute that to this optic system that Solaris is using. You can really control the amount of light that's going into the eye. You can position the electrode and stimulators very well, and that increases the reliability of the signals. And so you can see they're very nice looking. There's the intensity response functions. Here are the OPs, which are automatically isolated by their software, and we can do various things with these OPs. We can measure them individually or collectively. We can do Fourier transforms to look at the spectral frequency distributions, and so on. So this is a measure of how the ganglion cells function as well. Our light adapted ERGs, you know, in this particular example, they're a little bit noisy, but they're still very good and still very robust. We will try also to go after the photopic negative response, but that's a difficult one to get in the mouse, I think, in my experience. But importantly, we're adding the pattern ERGs to this study, and these are really beautiful, again, symmetrical pattern ERGs. The different lines are for slightly different intensities near saturation of this response. They're not very big, but they're very clear, and you can easily plot pick out the peak components. Whoops, let me go back one. Here are some preliminary results. The flash ERG, in fact, does show some effect, again, in the outer retina. The experimental animals show lower Vmaxes under dark adapted conditions. The same is true for the cone function. And again, you can see that the P1 for the control, P1 for the experimental animals is lower than the control animals, not so much for N2. But again, we only have a few animals here that are integrated here. We need more animals. In this experiment here, we were just trying to optimize conditions and test reliability. Group 1 and Group 2 are exactly the same animals. I mean, they're different animals, but same background. And we're looking at different spatial frequency of the stripes. The thicker the stripe you make, the bigger the response. But you can see that from group 1 to Group 2, you get highly reliable results. Again, for the larger check sizes, larger stripe sizes. This is the timing. There's a little bit more variability. And N2 also, again, you can see that I mean, this is pretty good given the order of magnitude of the signal. So that's the second project. The third project, which is my home base, has to do with looking at the recovery kinetics of the visual cycle. Some of you may know that when you bleach, you need to recover sensitivity by recycling all transretinol in the retina back to 11-cis-retinal, and that involves the visual cycle. So slow dark adaptation has been observed in a number of clinical diseases like AMD and in Stargardt's disease. Most of you, if you have any clinical experience at all, will know when you ask a Stargardt's patient if they have trouble dark adapting from the light of a midday afternoon into the darkness of a theater, they'll say, yeah, it takes me a lot longer. So we are studying those kinetics. The slow dark adaptation kinetics are also observed in some therapeutic interventions that we are using. For example, in Stargardt's disease, one of the things that we need to do is reduce the availability of vitamin A. And so we're looking at the effects of those restrictions on dark adaptation kinetics, and also slow dark adaptation is observed in some other diseases like cystic fibrosis, where they're being treated with fenretinide, which is known to slow the visual cycle. So the basic protocol for these studies is to present a bleaching light, and the Solaris does have some protocols already in place for this. We've modified it a little bit, but basically you bleach the photopigment from the retina, and then you use a probe using the ERG, with the ERG to track recovery of sensitivity as the eye dark adapts. On the right here is a psychophysically determined dark adaptation function. Bleach stops here. This is in a human, just for comparison purposes. You get a first, as you dark adapt after a bleach, your cones do the job first, and then your rods kick in and they increase the sensitivity. So can we do the equivalent of this in the mouse? And the answer is yes. And here's an example using the Solaris. For this particular example, we're using a very dim probe flash in order to probe recovery. This is in the linear range of intensity. It's not designed to saturate the A wave, for example. But here we take sort of the baseline. This is what the baseline response looks like. Then we present a bleaching light. In our case, it's 500 lux for 30 seconds, which takes out about 40% of rhodopsin. And then we watch it recover every minute over the course of up to 20 minutes. And so then we can plot the B wave, and you can see the B wave is shown here for different animals, but notice the wild types, they start at, there's no signal, you see it here, and then they slowly come back to the baseline amplitude. This is very much like that. And in this graph here, I'm showing you different animals, wild type. I have a couple of CFH mice in there. These are complement factor H mice. There's no physiological basis for CFH to have an effect, but our AMD patients do have these mutations and also have slowed dark adaptation we're looking at, and they don't have any difference in wild type. But the GNAT2s, which have no cone function, are slower, which suggests this is a mix of both rod and cone mediated dark adaptation. You can't see you can do the same kind of analysis using the A wave, but it's pretty sloppy because it's a dim probe flash and there's not much of an A wave, particularly at the low intensities. But if you jack up the probe flash to a saturating intensity that saturates the photoresponse, you get some very clear signals in the A wave. This is an example where we're having, this is the A wave. This is an animal being treated with isotretinoin, which disrupts the visual cycle, and you can see that it's slower recovery than the wild type. This is in an ABCA4 knockout mice, and you can see the same pattern, except these guys are a little bit slower than these guys. And here are the B wave responses that I just showed you. And you can go the opposite direction and ask what happens if you, instead of restricting vitamin A, which is what you're doing with inhibitors, vitamin A inhibitors, if you enhance vitamin A supplementation and provide more vitamin A, you can see the opposite happens, that it's plotted upside down, but basically recovery here with vitamin A supplementation is increasing, so relative to control. So these are some of the, I'm going to stop there because these are 3 projects that we're working on now. We're trying to integrate the Solaris in all of our studies to service all of the needs of our basic scientists in all of the divisions in our department. And that's a heavy order, and we think the Solaris can do that. I want to personally thank Eduardo in my lab for collecting all the data so far. He's a real expert. So that's it for me. I'll turn it back to you, Jeff.
All right, thank you, Steve. Excellent. Very, very nicely done, and 3 very interesting projects. Something I forgot to mention at the outset is Dr. Nusinowitz started using Solaris about a year ago, maybe just a little bit more than that. And obviously with the COVID shutdown, you can see his team has been really productive in just a few months using the system. So let's see, there's a couple of different ways we can use the last 14 minutes of the time that we have. The first option is to see if there's any questions that we have. So let me check with Natalie and see, Natalie, are there some questions that we should address first?
No, so there were only a pair of questions. The first was whether the touch-touch protocol allows for obtaining an ERG from both eyes. And the answer is yes. And what it does is it will typically interleave flashes. And what that does is if you assume that you want to wait, say, 10 seconds in between flashes, what you'll do is you'll set the interstimulus interval to 5 seconds instead of 10. And then what it'll do is it will flash the right eye, wait 5 seconds, then flash the left eye, then wait 5 seconds, and then it will flash the right eye. And so what you wind up doing is waiting the full 10 seconds between stimulus for any given eye. But you'll still have interleaved one flash from the other eye as well during that time. So it doesn't actually double the amount of time taken. It just adds 1/2 of your interstimulus interval. When you're doing a flicker, I'm pretty sure the video here is of a flicker. Since you can't have a flicker going on both eyes and use each other as a reference, what it'll do is it'll record the full stimulus for the right eye, and then it will do the full stimulus for the left eye. But you can use touch-touch to record both eyes. And what we've actually found is that the responses that you get from a touch-touch protocol, Jeff mentioned this a little bit early on, are far less variable than responses that you get if you use a third reference electrode in the forehead or elsewhere, like the cheek or something. And I actually have, that was pretty much the only question. If nobody else has any questions, I can actually show you a test that we did where we had one eye use, the right eye use the left eye as a reference, and the left eye use a needle electrode in the forehead, and we can show you the difference in variability.
Can I just show one thing here?
Yeah, absolutely.
These data here are all collected with the touch-touch protocol, and they're beautiful signals.
Yes. Catherine, I would say, so question in the chat, is the touch-touch protocol as sensitive as using a needle reference? And for VEP recordings using the Solaris, would you place a needle electrode at the base of the skull as with a traditional ERG? So yes, for VEP recordings, you would have the active electrode at the base of the skull as either a needle or a skull post, however you're used to doing it. But for the touch-touch, yes, it is as sensitive as using a needle reference. And then another question, could you explain the bleach recovery in detail again? So what the bleach recovery does, you start with a dark adapted animal, and you take a basic rod response from the animal. And then what you do is you bleach it. You do a very, very, very bright flash, or not flash, but a very bright background for some amount of time, a couple of minutes, 30 seconds in this case. And then you try and record a rod response 1 minute later, 2 minutes later, 3 minutes later. And you can watch the rods start to come back to a dark adapted state. So basically you just keep doing, you do a baseline rod response, and then you light adapt them, and then you try and look at a dark adapted response again, and watch them as they start to dark adapt.
Eduardo in my lab has set it up so that it's all automated. He doesn't have to do anything except click go. And he gets his baseline, and then the flash, the background light comes on, and then at timed intervals determined by the Solaris. I mean, we tell it what to do, but the Solaris does it automatically and goes all the way up to the end. It's a very easy protocol.
The other point then that could be added into this, as Steve mentioned, we have a basic bleach recovery protocol that comes with Solaris and the full field stimulators. One thing that Diagnosis tries to do is, to the extent that any of our customers are optimizing or changing protocols or creating their own new protocols, and they're willing to share those with Diagnosis so we can share those with other customers, that's a big part of what we try to do. It's up to each customer to decide if they want to opt in on that. But as much as people want to, then we try and just be a nexus of sharing either optimized or kind of derivative protocols for different purposes. And Steve alluded to the fact that that's part of what he's doing in his research. And that's something we love to help out with. And then to the extent we can share some of those learnings with others through protocols, so we definitely do that.
I'm certainly willing to provide some guidance. It's tricky when you do these bleaches because you have to battle the problem of phototoxic damage to the retina. If you use too much light for too long, then you'll never get recovery. You'll have damaged the retina, and there will never be any recovery. So it's a tricky protocol, and I have some publications on it that will guide you if you need it.
Excellent. Thanks, Steve. Natalie, did we have any other questions come in? And others, feel free to toss other questions in if you may have them.
No questions. So do you want me to really quick show the touch versus active reference ground in the same animal?
Yep, we can do that. And then there's a couple other things we can do right after that. Do I need to do anything here to give you control?
I don't have to do anything. I'm gonna make myself.
All right, well then it's better for me not to then.
So, all right, am I sharing my screen now?
I can see it.
All right, so what we're looking at, so what we're looking at on the top is the right eye, which is set up as a touch. So the right eye is using the left eye as a reference. And the left eye down here at the bottom is using a needle electrode as a reference. And so what we're looking at here is the average of 3 different sweeps for each eye. And you can see that while the average winds up being pretty much the same, there's a lot more variability if you're using a needle reference. And this is true at 0.01 candela per meter squared, 0.1, again, the top is the touch, and the bottom is active reference ground, 1 candela per meter squared. And then we added a couple more. I think this is 3, and this may be 10 candela per meter squared. Yes, so this is 10 candela per meter squared. So you can see that the touch paradigm gives a much greater reproducibility than the needle reference does. And they both wind up giving you a very beautiful looking, clean response. This is just an average of 3 sweeps apiece. But the touch just gives you a much greater reproducibility across individual traces.
That's excellent, Natalie. And I skipped over one other thing. Let me do a quick add-on to that. Can you see the charts?
Nope, I'm actually back as the presenter.
Okay. Let's make one other point on our amplifiers. Let's see. Get back to one of the system. This might be the best one. It's a little bit hard to see. You can verbally talk about this. So there's 5 input ports to the amplifier, 1 for ground. So you do have 4 input ports for the amplifier itself. So touch, most of the customers that use Solaris think have the benefits that Natalie just mentioned. But again, to emphasize, the amplifier has 4 ports, so there's a lot of different ways that you can use additional electrodes for VEP, as Natalie mentioned, or more traditional reference needle electrode setups as well. So it's not a limitation of the system architecture. It's taking advantage of the light architecture, as Steve said. Okay, we still have a few more minutes. One more chance for questions. Maybe as we're doing that, Steve, do you have any thoughts, short of retiring in our COVID environment, as you hopefully jokingly mentioned earlier today? Any thoughts that you have in terms of additional processes, procedures, test types that you hope to be able to implement on Solaris over the next year? Beyond things that you
yeah, it depends on the demand characteristics of the scientists around here. You've shown examples of, for example, the C wave, which is RPE driven. Some who are doing RPE research might be interested in doing using that modality, and anything that we could possibly do. The only thing that you haven't done, but I think you can do it, is to do the 2-flash paradigm. That, remember, we published that probably 15 years ago. Me, David Birch, and Don Hood, and David Pepperberg doing the 2-flash to isolate the in vivo rod response. And I'm pretty sure you guys can do that, but we have not done that yet.
We haven't done, and we have not yet either. It's a good example of something to work on yet together. And the other thing I would add on that Steve has pointed out, some of the intensity response graphing and coming up with function parameters that he showed in his charts, he does those offline from our software. All of the test is taken in the Diagnosis Solaris software. The analysis that leads to parameters like Vmax is currently done outside of our system, but those are things that Steve has pointed out to me would be good if we can work into our software, and that's a good example of something we hope to bring into our software here in the coming year as well. So always trying to look for additional enhancements, protocols, or some of the basic analytical techniques that we can work into our software.
We have,
you know, I've had some strange ideas over the years, and I know that Diagnosis has been very cooperative with me, starting way back when Bruce made my first notch filter. This has got to be 25 years ago. So, you know, one of the things that I really like about Diagnosis is the customer support and their willingness to try to develop things with you. It's obviously one hand washes the other. It's good for us, it's good for them, but they're willing to help.
Excellent, thanks, Steve.
we do have a couple more questions in the chat. Steve, I know you've got a hard stop in about a minute. We were asked, what are the programs used to fit some of those functions you described, MATLAB or similar? What do you use?
MATLAB, we used MATLAB for the A-wave fitting, but there are a number of software packages that you can do the B-wave fits. And what do we use? Hold on one second and I'll tell you in a second.
Well, you figure it out. We export raw data to in ASCII in either to the clipboard or in a CSV or TXT file. So almost anything that can import a CSV or TXT file or accept a clipboard paste function can accept it. So go ahead, Steve.
We also use GraphPad. GraphPad is an easy software package. It's relatively cheap, and you can type in your own equations, your own formulas, and it'll do the best fit for you.
And then we had another question about how the system performs with rabbits.
You want to cover that quickly, Natalie, before we close up? Just 30 seconds.
We do have a rabbit table that makes the basically extends the reach of the Solaris, because rabbits, particularly the New Zealand whites, can get very large. And we don't use the the same stimulators for rabbits. We have ones that look a little bit more like sort of photographer's lighting lamps. We recommend using electrodes with those. But we do have rabbit configurations, and they work quite well. And then we had one other question about to what extent does the needle reference vary depending on the size of the rodent? And that placing a forehead needle electrode is pretty challenging in very young mice. So I'm going to show you a photo again of a 21-day-old mouse getting an ERG from a Solaris, just using touch. And we got a great response there with this animal. So you don't need to use a needle electrode for most protocols using the Solaris, and we do find that gives you a much better response.
Excellent. Great questions. Well, let's wrap up. Again, Steve, thank you for joining us and sharing the research projects that you and your team are working on out at UCLA. Very much appreciated.
My pleasure.
Excellent. And thank you, everyone, for joining us today. We may have future Solaris webinars. If we do, they'll probably be in a couple of months, perhaps in September. So feel free to follow up with the diagnosis sales team if you have any other questions. And if you have questions for Dr. Nusinowitz, he kindly shared his email address in our chart as well. So thank you again. Bye-bye.
Bye.
Thank you very much.